each of the analytes. And you can see the intra-assay, which is the between spot with a minimum of 4 spots per analyte, compared with the inter-assay CV, which is between biochips and 5 biochips read on a single instrument, compared with the intra-instrument with 3 instruments using a total of 13 biochips. So the CV data here is very respectable data. In our comparison of within day and day-to-day CVs, we see that we're able to confirm and actually do a little bit better than DBI reported on that previous slide. Here we have a high control sample that is run in replicates of 5 on the same day for the within run, and run in duplicate on each of 4 days for 8 determinations on the day-to-day CVs. So again, highly respectable precision data. This slide shows the linearity, and again, this is spiked human plasma, serially diluted by 2-fold, and then the recovery is calculated. It's a little bit challenging data here because it's hard to find one serum sample that has all these cytokines elevated. So that's why we had to use the spiked plasma samples. And you can see here that the recoveries are really quite good through at least the 1 to 32 dilution. You can see some drop off here at the 1 to 64 dilution, but even when this data is included, we see that the mean recovery for all of these analytes is really quite surprisingly good. And again, one of the things that we're doing is reassessing the normal range for all these analytes. Not really a clear requirement, but something we think should be done. So we are in the process of doing this. This is the first 13 samples that we've analyzed, and the normal range data are consistent with data reported in the literature. One of the issues that we have seen with multiplex protein assays is the interference of one assay system with the other. And so the turning point, I think, in my acceptance of this assay was this type of interference data. So here what we see is the analyte measurement with the spike concentration coming here. And so we're spiking each one of these to very high levels. And you can see that looking at this range of recovery values, that indeed the values are really quite consistent, showing no significant cross talk, if you will, between these various antibodies. So unlike our experience with platform, these particular set of antibodies are working quite well, and hats off to Decision Biomarkers for quite excellent antibody selection in avoiding this type of crosstalk interference. This again is the set of standard curves for the angio chip, which is ICAM, the intracellular adhesion molecule, E-selectin, the tissue inhibitor metalloproteinase, IL-8, the angiopoietin receptor, hepatocyte growth factor, fibroblast growth factor, platelet-derived growth factor, and the vasoendothelial growth factor receptor, and the ligand. So again, notice the log range going 2 to 3-fold for each of these with nice response curves. So very good standard curve set. And this is again some of the assay parameters for the angio chip. Again, each one of the analytes in this chip, very good dynamic range in the picogram per mil category there in units, with excellent sensitivities. Again, with sensitivity defined operationally by the mean value plus 2 SDs of 10 replicates of the zero standard. And again, precision studies, again showing the within day and day-to-day precision, again very respectable CVs for the most part, and certainly acceptable for the drug trials that we're planning. Again, linearity, again showing again spike samples with dilutions, again showing excellent recoveries for the most part through each of these analytes, and again where they're not totally 100% recovery, quite consistent over these ranges. So we feel that that the data that we generate from this will be highly reliable. Again, normal range studies in place, and again showing again those 13 controls showing very acceptable and correlative to previous published data. So what we can say in conclusion is that DBI has developed 2 automated multiplex immunoassays for the cytokine 8 angiotensin chip for the AVANTRA. The assays use biochips containing all the necessary reagents and controls to ensure easily generating reliable data. The platform is easy to use, and the automation allows for minimal hands-on time for the operator. In fact, we have 1 operator using 4 machines. So it's easily addressable from the high volume throughput. You just got to add more machines. We are independently evaluating these data as required by CLIA, as you all know, and we intend to operate these in our clinical drug trials at MD Anderson. Now, we just finished a study for GlaxoSmithKline, in which we demonstrated, along with their help, that 6 of these markers, I think 4 from the angiogenesis and 2 of the cytokine markers, were predictive of patient outcome. So we've actually demonstrated, and this data will be presented soon at an upcoming meeting. In fact, I just got an email to get the author disclosure in today, so it'll be presented soon. But again, I see no problems from the laboratory standpoint in offering these assays, and I'm encouraging Decision Biomarkers to proceed with haste to develop other biomarker chips for us, maybe particularly in the bone marker mode. So again, I think this is a highly reliable system, and hopefully we'll have this data published soon.